Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Charcoal selective medium. Principles, uses, and limitations in Campylobacter detection. Table of contents. Introduction. Composition. Principle. Preparation. Result interpretation on charcoal selective medium. Uses. Limitations. Introduction. In 1984, Bolton et al. Suggested that charcoal could be an effective substitute for blood in culture media used for isolating Campylobacter spp. This claim was later validated by Entz et al., who confirmed that the isolation rate for Campylobacter was higher when using the charcoal selective medium. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Let's take a look at the composition of charcoal selective medium. It starts with 10 grams of beef extract per liter, providing essential nutrients like nitrogen, carbohydrates, and vitamins. Next, we add 10 grams of gelatin peptone, contributing amino acids and peptides. Then, we include 5 grams of sodium chloride for electrolyte balance, helping maintain osmotic equilibrium. There's also 3 grams of casein peptone, another source of amino acids. To enhance aerotolerance, we incorporate 4 grams of charcoal, a key component, along with 0.25 grams of ferrous sulfate and 0.25 grams of sodium pyruvate. These ingredients work together to reduce oxygen tension and help the bacteria thrive in aerobic conditions. For selective inhibition, 1 gram of sodium desoxycholate is added. This helps prevent the growth of unwanted bacteria. We also include antibiotics, 100 milligrams of cyclohexamide to block yeast and mold growth, 32 milligrams of ceftriaxone to suppress gram-negative enteric bacilli, and 20 milligrams of vancomycin to inhibit many gram-positive bacteria. Hematin, at 32 milligrams, and 12 grams of agar are added to support bacterial growth and help solidify the medium. Finally, the pH is carefully adjusted to 7.4, with a tolerance of 80.2, measured at 25 degrees Celsius. Principle of charcoal selective medium. The medium contains beef extract, which provides essential nitrogen, carbohydrates, vitamins, and other nutrients necessary for the growth of Campylobacter. Gelatin and casein peptones contribute amino acids and peptides as additional nutrients. Charcoal, hematin, sodium pyruvate, and ferrous sulfate enhance the aerotolerance of Campylobacter species. These supplements are believed to act as quenching agents for photochemically produced toxic oxygen derivatives. Charcoal serves as a detoxifying agent and reduces oxygen tension. Sodium chloride supplies crucial electrolytes, maintaining osmotic equilibrium and preserving cell integrity. Sodium desoxycholate is included as a selective agent to inhibit certain bacteria. Ceftriaxone, a cephalosporin antibiotic, suppresses the growth of gram-negative enteric bacilli and some gram-positive species. Vancomycin, a glycopeptide antibiotic, inhibits many gram-positive bacteria. Cyclohexamide, an antifungal agent, blocks the growth of yeast and mold. Preparation of charcoal selective medium. Add the components to distilled or deionized water and adjust the volume to 1,000 ml. Mix thoroughly, then gently heat the mixture, bringing it to a boil while stirring frequently. Autoclave the solution for 15 minutes at 15 psi pressure and 121 degrees Celsius. Allow the solution to cool to 45 to 50 degrees Celsius. Pour the medium into sterile Petri dishes or distribute it into tubes. Shake the flask while dispensing to ensure the charcoal remains in suspension. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Result interpretation on charcoal selective medium. Campylobacter jejuni produces two distinct types of colonies. Small, raised colonies that are grayish-brown, smooth, glistening, with a clear, translucent edge. Flat colonies that are mucoid, translucent, grayish, and have an irregular edge. On fresh medium, colonies may appear flat, irregular, or spreading. Some strains may form a thin film on the agar or develop colonies that extend along the streaking line. On less fresh medium, colonies are typically 1 to 2 mm in diameter, round, convex, and glistening. These colonies can range in color from yellowish to gray or pinkish and are non-hemolytic. Uses of charcoal selective medium. It is utilized for the selective isolation and presumptive identification of Campylobacter species from food and human fecal specimens. Limitations of charcoal selective medium. It is recommended to perform biochemical, immunological, molecular, or mass spectrometry testing on colonies from pure culture for comprehensive identification. Extending the incubation period to 72 hours may enhance the isolation rate of Campylobacter. Campylobacter coli, Campylobacter fetus, and some strains of Campylobacter jejuni may be inhibited by cephalosporins. Campylobacter jejuni should be incubated at 42 degrees Celsius, as it is a thermophilic organism. The higher temperature provides selectivity by inhibiting accompanying microflora and promoting the growth of Campylobacter jejuni. Agents in selective media might inhibit some strains of the target species or allow the growth of species that the medium is designed to inhibit, especially if the species is present in high numbers in the specimens. Therefore, specimens cultured on selective media should also be cultured on non-selective media to obtain additional information and ensure the recovery of potential pathogens. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!